Hey Team Bio, welcome to your screencast, an introduction to osmosis and diffusion. So we're going to be talking in like way zoomed out perspective, how things move into and out of cells. So first of all, let's talk about what these two words mean. Let's start with diffusion. So diffusion is the tendency for particles of any substance to spread out. And this happens pretty quickly if we're talking about a gas, a little bit more slowly if we're talking about a liquid, and even more slowly if we're talking about a solid. But eventually, particles spread out. All right, I'll get to that in a second. Okay, so it's the tendency for any substance, for particles of any substance to spread out. The movement of individual molecules is random. But on net, they move down their concentration gradient. So when we add up like the movement of particles going left and particles going right, if we start out with more particles on the left than on the right, then over time, there are going to be equal numbers of particles on the left and on the right. On net, they've moved down their concentration gradient. Until they reach a property that we call dynamic equilibrium. When this happens across cell membranes, we call it passive transport. And it's called passive transport because it requires no energy input from the cell. Okay, so osmosis is a special case of passive transport. It's the diffusion of water. Specifically, water across a semi-permeable membrane. So we talked a little bit, we haven't really talked that much about this, but this idea of semi-permeable means the membrane lets some things in, but it doesn't let other things in. So for now, what you need to know is that the membrane allows water to cross. but it prevents glucose from crossing. Okay, so osmosis is our very special case when water moves across the cell membrane, the semi-permeable cell membrane, which allows water to go in and out, but it prevents glucose from going in and out. Okay, so quick review from methods. A solution is when you have a solvent, which is the liquid, plus a solute, which is the dissolved particles. So the solvent in our case is always water in biology, almost always water, and the solute in this case is glucose. So we're going to be looking at a couple different solutions when a solution on the inside of a cell is different from a solution on the outside of the cell and what happens. Okay, we've got our first situation. So quick key, I know this didn't show up very well in your diagram, but these pink dots are glucose, and the blue highlight is water. So in this situation, you can see that there's a lot more glucose inside of the cell than outside of the cell. We call this a hypotonic solution. So a hypotonic solution has more glucose inside. Okay, so in this situation where there's more glucose inside, the concentration of the solution inside is greater than the concentration of the solution outside, what's going to happen is that this solution wants to be in dynamic equilibrium with equal concentration of glucose to water inside and outside. Glucose can't move across the cell membrane. Only water can move. So what happens in this situation is that water rushes into the cell. Sometimes the cell just grows in size, but if the difference in concentration inside the cell and outside the cell is too great, what can happen is a situation where the cell bursts. So I'm going to draw that here, kind of like a balloon. So the cell, actually, but I should draw it a little bit bigger because the cell would first expand and then it would burst, and water and glucose would rush out, and at that point the concentration would equalize inside and outside the cell. So a hypotonic solution can cause a situation called cytolysis. Cyto referring to the cytoplasm, the inside of the cell, and lysis is to break or to burst. So hypotonic solutions can cause cytolysis in some cases. Here is the opposite situation. Here we have more glucose outside of the cell than inside of the cell. This is called a hypertonic solution. Again, glucose can't move in to balance out the concentration. The only thing that can happen is that water can move out, which is exactly what happens here. So the cell shrivels. So here water rushes out and the cell shrivels. This is called plasmolysis. Okay, the last situation is when we have an equal concentration of glucose inside of the cell and glucose outside of the cell. So this is equal glucose in and out. So what happens here is that water still moves across the cell membrane. It still goes in, but it also moves out at the same rate. So water here is moving into and out of the cell at the same frequency. This is what we call dynamic equilibrium. So water is still moving in, but water is also moving out. It's moving in and out at the same rate. And as a result, on net, when we sum up all this inward movement and all this outward movement, it's zero movement on net. So this is dynamic equilibrium. Water's still moving, but the concentration of the glucose is not changing relative to the outside, because inward motion and outward motion are balanced. So here we have a cell that stays the same size. Okay. If you flip over the page and look at the next side, you're going to see a couple different situations. So animal cells and plant cells enjoy different conditions, and this has to do with their structure. So animal cells have no cell wall. As a result, if you put an animal cell into a hypotonic solution, into a situation where there's more glucose on the inside of the cell than on the outside of the cell, water's going to rush in. So it's this top situation, and it will burst open. So for example, imagine you had a person who's extremely dehydrated. If you just injected water directly into their veins, you would be adding a lot of water, but you would also be exposing their cells to hypotonic solutions, and you would really quickly kill them because all their cells would burst. Okay, so when someone gets extremely dehydrated, instead you hook them up to a salt water solution, and that helps rehydrate them. It adds more water to their system, but it also creates a situation where their cells are always in an isotonic solution. So animal cells prefer, or maybe not even prefers, that's too weak a word, demands isotonic solutions. They want to have conditions where the inside of their cells matches the outside of their cells in terms of solute concentration. Okay, if you put an animal cell in a hypertonic solution, it will plasmolyze or shrivel like this. Okay, a plant, however, has a cell wall. And the cell wall is really structurally stable, and it prevents the cell from bursting. In any conditions. So plants actually prefer to be in a hypotonic solution. They like to be in a situation where there is always water pressure on the outside of the cells pushing to get into the inside of the cells. This creates a state called turgid, and the plant is happy and very upright. When a plant starts to move from a hypotonic solution to an isotonic solution, where the concentration of solution inside and outside of the cell is different, is the same. Now there's not as water pressure going in is the same as water pressure going out, and the plants the cells become flaccid and the plants start to wilt. In a situation where a plant is in a hypertonic solution, it's extremely unhappy and it actually pulls away from the cell membrane. You can see these gaps between where the cell membrane is attached to the cell. Sorry, I said pulls away from the cell membrane, but what I meant to say is the cell membrane pulls away from the cell wall. So you can start to see these gaps between the cell wall and the cell membrane where the cell is pulling away. So a plant here is just kind of wilted. This one would be extremely sad, extremely wilted. So plants and animals would like to be in different conditions. Animals demand isotonic solutions. They'll shrivel in hypertonic solutions, and they'll burst in hypotonic solutions. Plants love hypotonic solutions. This is when they're most happy. They're upright. It provides them structural stability and rigidity. And when they're in isotonic or hypertonic solutions, they look wilted and they look like they need to be watered. Okay, that's all.